to today's teaching point about cell sorting. There are 4 different commercially available solutions for sorting and isolating cells. These include magnetic sorting, electrostatic sorting, physical mechanical desorting, and of course the new microfluidic sorting. Today we're going to focus on magnetic sorting and electrostatic sorting and how those two can work together. Let's start off with a review of magnetic sorting. So magnetic-based sorting involves using a magnetic bead that is coupled to an antibody. Those antibodies label cells. We place those cells in a magnetic field. The cells that have been labeled the antibodies are attracted to the magnet, while the cells that are unlabeled remain in the supernatant. We are able to isolate 2 populations, those unlabeled and the labeled cells, when we remove the labeled cells from the magnet. Now, electrostatic sorting can either be done using jet and air or Cuvette. And the difference is, where is the laser intercept? In a Cuvette-based system, the laser intercept is here in a Cuvette. This improves the light collection compared to jet and air sorting, but both principles are the same. A cell is identified in a stream, which is vibrating at some frequency. It's going to pass through a nozzle. This is going to contain the stream, and we're going to see the cell here at the very bottom of this. This is our breakoff point. This is the last point where between a stream and the droplets. We charge this stream so that the droplet with our particle of interest obtains an electric charge. As that moves off, becomes this first droplet, we turn off the electric field. So now this drop is charged and the drop here is not. The cell in the droplet continues down into an electric field where it is deflected to our collection tube. It's important to remember that electrostatic sorting, cell sorters sort droplets, not cells. Now, this nozzle is important because this nozzle establishes the pressure, which establishes the frequency, which establishes how many events per second we can sort. This nozzle is based upon the size of the cells we're going to sort. Here is a list of common cells that we sort along with their volumes. Using the famous 4/3 pi r cubed, we can calculate the rough diameter of these cells, and based upon the rough diameter of the cells, pick a nozzle that we'd like to use. And we typically want a nozzle that's 4 to 5 times the size of our cell. So you can see here something for an alveolar macrophage probably could sort in the 100 micron, but it's better to sort at the 130 micron. The nozzle diameter affects the sheath pressure, which affects the frequency. Here we have a table from Arnold and Lanigan showing nozzle diameters, approximate sheath pressure ranges for each of these diameters, and approximate frequency of sorting. So you can see for a 70 micron nozzle, we have a sheath pressure between 45 and 60 psi, which means we can have a frequency of 65 to 100 kilohertz. This will become important in just a minute. You'll notice as we go to the higher diameter, sheath pressure goes down, as does frequency. Another consideration we must make is how we threshold. Now, on analytical systems, we put our thresholds up pretty high so we can eliminate things that we don't want to see in our data set. In the case of cell sorting, we want to keep the threshold as low as possible. Here we have 3 different beads at a threshold of 10,000 events per second. Here we show 3 different beads with a threshold of 10,000 scatter or 50,000 scatter. We've identified a small bead here, a bead that we're going to sort, and then this extra bead up here. Now, at the high threshold, we don't see these small samples. So if we take a look at this 10K threshold, our pre-sort, we have about 28% of the events in this population and about 58% here. I'll show you the post-sort analysis in a minute, but I'll give you a heads up. We get 97% purity of these cells with a little bit contaminating. Now, if we look at that with the 50K threshold, there's no events in the small gate, 85% in this gate, but after post-sort, and when we turn the threshold down to visualize those events, we now see that about 11% of the events are in this small gate. The flow cytometer was blinded to those events, and so they can contaminate our sorting population. This can be a problem if these contain some compound that might be impacting your downstream analysis. The other thing we want to talk about is the event rate. How quickly do we sort? Now, when we think about this, we're actually going to look at Poisson statistics, which are a measure of the probability of an event appearing, and with all other events being independent. So if we assume having 1 droplet with 1 cell, we can see that there's a 37% probability that the droplet will be empty, and a 37% probability that it'll have 1 cell. But notice there's an 18% probability that it'll have 2 cells. Now, if we go to one cell every other drop, we can see that frequency of singles drops down to 30, but we still have about an 8% probability of having two cells in one droplet. If we go to one droplet, one event per 4 droplets, we can see we drop down to about 19% that are single, but only 2% that might have 2 or more. Going up to one drop or one cell for every 6 drops, we still have about 14%, but we still have about 1% that contain 2 cells or more. Based upon these numbers, we want to sort with an event rate of 1 cell per 4 droplets. So here's a chart showing the nozzle size, the rough frequency of droplet generation, and the maximum number of events per second. Based upon this, you can calculate the number of cells per hour that can be sorted and a recommended concentration per mil that you might want to bring your sample to the sorter in. Now, if we take all this together, the other factor that we have to think about is the frequency of the population. So if we have a downstream application that requires 100,000 cells, we can look at the effect of the varying the frequency of population, the recovery of our source sorter, the recovery of our sample processing to determine the approximate number of cells that we're going to have to stain. And based upon different frequencies, we can then figure out how long it would take to sort that 100,000 cells. And you can see as frequency decreases, the number of cells we have to stain increases, the time on the sorter increases. As our recovery of our sorter decreases, more cells we have to stain, the longer it will take. This is typically going to happen if you've got a sample where you're doing a very tight single cell analysis where you want to sort just a single cells and make it very, very clean. And of course, if we work on optimizing our sample recovery, typically we lose between 20 to 30% of our cells through the processing, depending on how many centrifugation steps that you have. So you can see the shorter our recovery, the more events we're going to have to sell. More events we're going to have to stain. Let's take a look at an example why we might want to combine depletion with cell sorting. And that would be in a case where we had a very low frequency, say 0.1%. So we're going to have to stain 250 million cells. Well, if we think about that, if we were to stain, in this case, 200 million cells with a 0.1% target, it's going to take about 2.5 hours to sort. Now, if we do a depletion with beads, as shown down here, we can eliminate potentially 90% of those cells, meaning that we now have a 1% target population. These types of depletions take about an hour or so. Now you've got a 20 million cells, which you can sort at 22,000 events per second. It's going to take about 15 minutes of sort time. You've saved 1.25 hours by doing this depletion, which is going to improve your sort because you've got a higher target population. Your cells are going to be happier because they're out there spending less time being manipulated. So if you have a very infrequent population or very rare population, a depletion study or a depletion experiment is a good way to eliminate a lot of those cells and reduce the time on the cell sorter. If we want to summarize what we've talked about today, the size of the cell matters because it dictates the size of the nozzle that can be used. The nozzle size impacts how fast the system can run and how many droplets per second can be generated. Based on Poisson statistics, as a rule of thumb, we want to sort at 1 event per 4 droplets for best results. You want to be mindful of setting your thresholds because if they're too high, this will impact your purity, especially because the system is blinded to things be mindful of thresholds because if they're too high, this will impact purity because the system is blinded to those smaller events, which may contain contaminating cells that will impact your downstream results. Last but not least, consider pre-enrichment steps that will help speed up the time on the cell sorter.